this is the part number, I guess, fifth. And now in this, we are going to talk about spatial summation and temporal summation. So one is spatial summation and it is related to space. What does that mean? It means that, for example, you know that, so this is a postsynaptic membrane and these are the presynaptic membranes. So whenever a presynaptic membrane, it releases the neurotransmitter, a single presynaptic membrane can like give 0.5 millivolt. But you know that to raise this minus 65 millivolt to the level of minus 45 millivolt, which is an excitatory postsynaptic potential, which will take it to the threshold level and generate an action potential, we need 20 millivolts of positive potential. So we need such 0.5 multiplied by 20 makes it around 40. So we need at least 40 to 60 such neurons to fire at the same time to so that they reach the 20 millivolt potential. That is how it happens. Now what do we call this? Here we can read that to reach the excitatory postsynaptic potential, each neuron or each presynaptic terminal can only fire 0.5 to 1 millivolt, and it does not reach the 20 millivolt threshold very easily. That is why different neurons work together and their firing is summed up simultaneously at the level of postsynaptic potential, and hence this spatial summation, that is at the level of space there are different very different kind of presynaptic terminals which are firing the neurotransmitters to raise the potential in a collective manner. For example, if we look at this 4611 figure, you can see that when 4 synapses are firing, then the threshold or the potential is rising only a little bit. If 8 are firing, it is rising a little bit more, but now 16 are firing. So what happens in such a state that you can go from minus 65 above to the level and reach the level of minus 45, and when you reach the level of minus 45, that is EPSP, then an action potential is generated. So this is the level of spatial summation, summing up all the neurons that are present in a single space. The next summation is the temporal summation, that is related to time. For example, if we said that whenever a presynaptic terminal fires, it fires within 1 or 2 millisecond, and this firing lasts for about 15 millisecond. So what happens is that if that neuron or presynaptic terminal, it fires successively, like you can say this was the presynaptic terminal or this was the presynaptic terminal, and it is firing a lot of signals at the same space within very small amount of time. So the difference between the time is very small, and as a result, if we look at this firing level on a larger scale, we can see that at this point, when we look at this at a macro level, we can see that a firing level is recorded at this level. So this is how the time or the temporal summation works. That same presynaptic potential or presynaptic membrane is firing different potentials but with a small gap of time, and this small gap of time, a large amount of action potentials being fired within a very limited amount of time creates a larger potential which can activate or reach the excitatory postsynaptic potential, and this summation is called spatial summation. Temporal summation. The spatial was related with space, that is meaning more number of presynaptic terminals, and temporal is related with time, meaning more action potentials within a very limited amount of time. So the words that they have written is successive discharges from a single presynaptic terminal. If they occur rapidly enough, they can add to one another and hence they can summate, and this is known as temporal summation. Now simultaneous summation of inhibitory and excitatory postsynaptic potential. What happens is that consider this a postsynaptic membrane, and on this postsynaptic membrane certain presynaptic membranes are doing the positive signals, other are doing negative signals. Rather, some are going to give them the EPSP, that is excitatory potential, the others are going to give it an inhibitory potential, but the final potential will be the sum of these. Like if this is giving some positive value, if this is giving some negative value, so they both will be added to give the final result, and that final result is going to act on this minus 65 millivolt, and this addition work is going to happen. So if IPSP tends to decrease the membrane potential to a more negative value, EPSP tends to increase the potential to the more positive value. So if a neuron is being excited by an excitatory potential, an inhibitory signal from another source can often reduce the postsynaptic potential. So that is why what we do is that we simultaneously summate the inhibitory as well as excitatory potential and get the final result by the algebraic addition of both the potentials. Now, what is facilitation of the neuron? Sometimes the membrane is having positive potential, like sodium ions are coming to it, but the potential is not enough to reach to the threshold level. So what happens that the nearby neurons, they facilitate them by diffusion of the sodium ions. The nearby neurons, they give those sodium ions to the neuron that is trying to reach the threshold level, as a result of which we call this facilitation of the neuron. Side or neighboring neurons are helping one neuron to reach its threshold potential so it can fire or it can generate an action potential, is the facilitation process. Now, if you know the structure of the neuron, you always know that the neuron has a cell body, and above the cell body, the part which conducts the signal towards the neuronal cell body is known as dendrite, and the part which conducts the signal away from the cell body is known as the axon. So now they are saying that dendrites, that is the portion which is going to conduct the signals toward the cell body, it is very large, like it is having a large area. It is spread over an area of 500 to 1000 micrometers, and it gets all the signals from that area. And now they are saying that if there is presynaptic terminal, so 80 to 95% of the presynaptic terminal will synapse with a dendrite, whereas 5 to 20% will synapse with a cell body. So major synapses are between the presynaptic axon and postsynaptic dendrite, but sometimes they can be with the presynaptic axon and a postsynaptic cell body, but that is very rare. Now, most dendrites cannot transmit action potential. Why they cannot transmit the action potential? Because they have few voltage gated sodium channels, and as we know, voltage gated sodium channels are necessary because they allow the sodium ions to move inside. When the sodium ions will move inside, the negativity will be reduced, positivity will be achieved, and the membrane will move towards the threshold. But if there are less sodium channels, the positive ions cannot move inside, the negativity remains over there, and threshold is increased. But now they are saying that there are some neurons that, even though they have few voltage gated sodium channels and they cannot reach their threshold very easily, but they transmit electronic currents. These currents, they travel from the cell body towards the axon and then travel to the synapse. Due to this spread of the current, they have very important role because they can stimulate or they can inhibit certain synapses. So these currents are also very important while we talk about the conduction in the neuronal cell body. So now we have this topic, which is decrement of electronic conduction. So what do we mean by decrement? Decrement is that sometimes the action potential that is being transmitted, it is lost somewhere in between. For example, consider this a neuronal cell body, and this is a dendrite, and the features that are written over here is that dendrites are long and they are having membrane that are thin. So whenever this dendrite is long and is having a thin membrane, what will happen? The chloride ions and the potassium ions can move outside. Potassium ions can move outside, chloride ions can move inside. So the excitatory postsynaptic potential that was coming it is going to be reduced. Like a positive potential of 10 was coming, so when it reaches the cell body, due to the outflow or efflux of the potassium ions and influx of the chloride ions, the potential decreases to like plus 3 millivolts. And this decrease in the potential due to this thin membrane and its efflux and influx principle, we call this decrement or decrease in the neuronal potential due to the fact that the dendrite length is very long. So this is known as decremental conduction. The farther the excitatory synapse is from the cell body, the greater will be the decrease or the decrement, and lesser will be excitatory signal and more will be the inhibitory signal. And whenever there is difficulty for the excitatory postsynaptic potential to reach, what will happen? Threshold will be increased, neuron will be difficult to excite. So whenever a dendrite is small, it is going to excite the cell body more, and whenever the dendrite and cell body connection is large, there will be a lot of decrement, and as a result of which the EPSP and threshold will be not reached and excitation will not be produced very easily. So now summation of excitation and inhibition in the dendrite. So this is a dendrite, this is the cell body, and this is the axon. So what they are saying that at the tip of the dendrite, there is a strong EPSP, excitatory presynaptic potential. So these are the excitatory ones, but when the dendrite reaches the cell body, there are inhibitory signals going over here. So the net effect will be summating the excitatory plus inhibitory effect, and when we will summate it, then we will get the final one, as a result of which we will get an action potential. Now what happens is that in the axon, sometimes the point where the axon is originating from the cell body, and at the level of axon hillocks there can be inhibitory synapses, and these inhibitory synapses, they are going to increase the negativity inside. When they will increase the negativity, that is, the potential will go even more downward from 60 to minus 70. It means threshold is difficult to reach. As a result, the membrane is difficult to excite. So this principle, that is at the level of axon hillock and the initial axonal segment, where the signal is actually formed, there where the action potential is actually generated, what happens? The action potential is nullified at that very point, increasing the threshold for excitation. So what do we recall from all of this? That whenever there is going to be excitation, it is going to be the sum of the excitatory and inhibitory potentials in the dendrite as well as in the axon. Now they are telling about the excitation state of the neuron. I've told you there is summatory effect whenever the EPSP and the IPSP, that is excitatory and inhibitory signals, are there. They are going to act collectively, and if the sum is positive side, then there will be excitatory state, but if the sum is on the negative side, there will be an inhibitory state and signal will not be produced. But now if it is on the excitatory state, what will happen? Action potential will be generated, signals will be fired repetitively. Now we'll move to this figure 46. In this figure, on this state, they are showing the threshold levels. On the other side, they are showing the maximum frequency. Now the threshold, which is most low, is of the neuron number 1, and threshold most high is of the neuron number 3. So you can see that the lower the threshold, the easy it is to excite the neuron. Second thing, highest maximum frequency. So now you know that whenever the neuron is high threshold, it means a lot of signals are required for it to reach the threshold, and when it reaches the threshold and it starts firing, so a large number of positive signals are there, and these positive signals, they will fire simultaneously, and as a result of which maximum frequency will be reached. As we can see in the neuron number 3, it has high threshold, but its maximum frequency is also higher. Now they are explaining that in the central nervous system, there are some neurons that are in the normal excitatory state. That is, they are excited at all the times. And why is that important? Because it keeps us awake, like the reticular formation. It keeps us awake at all those times. But whenever we are sleeping, this reticular formation stops working. So what happens? We can call this an inhibitory state, and in such a case we have to impose the inhibitory state and we have to open its inhibitory channels. So that is how we can actually control our brain functions, by controlling the activity of the simplest form of the central nervous system, that is the neuron. Now we are going to study about the special characteristics that of the synaptic transmission. One is the fatigue, second one is the effect of acidosis, alkalosis, third one effect of hypoxia, etc. So now what is fatigue of the synaptic transmission? So now, as we know, this is a presynaptic terminal, this is a postsynaptic terminal. So what happens is, whenever an action potential is coming, it is causing the release of neurotransmitters in this space. And if a large amount of action potentials are coming, a large amount of neurotransmitters are being released. But what happens? A point comes that when neurotransmitter reserves are all empty, and now you cannot excite the membrane, the postsynaptic membrane. This is the state of fatigue. So when excitatory synapses are repetitively stimulated, the postsynaptic neuron, first it is very great, the signal is very great, but soon fatigue starts to happen. Why? Because the membrane is overexcited. And due to this overexcitation, what happens is a large number of action potentials are being fired. So person becomes epileptic and the seizures are occurring. But whenever these neurotransmitter, they are all used up, what happens? A state of seize or stoppage in the cycle takes place. And that is why an epileptic person who was having a large number of seizures, his seizures stop after some time. Why do they stop? Because now the neurotransmitters have all been used up and the synapse has been fatigued. Thus, this fatigue is actually a protective mechanism against overexcitation. Now what happens is, now they're saying how many action potentials can be transmitted. There are 10,000 action potential that can be transmitted, and transmitters can be exhausted for about few seconds to a few minutes. Now how this fatigue process occurs is that that there is the postsynaptic membrane, and the postsynaptic membrane has a large amount of receptors. So first thing first, that these receptors are inactivated. Second thing was the neurotransmitter stores are all used up. Third thing is going to be that in the postsynaptic membrane, the you can say the level of the balance of the sodium, potassium, calcium ions, chloride ions, that will all be changed. So inactivation of receptors and abnormal concentration of ions. And another point that I added was the using up of all the neurotransmitter vesicles. These 3 points, they actually cause the fatigue of the synaptic transmission. Now we come to effect of acidosis and alkalosis. Your normal body pH is about 7.4, but whenever it increases, like there is a state of alkalinity, so it increases to 7.8 and 8. So person can have cerebral epileptic seizures, because the pH, when it increases, when it goes towards alkaline side, the person has a large excitation. Neuronal excitability is increased, and as a result of which, whenever there is overexcitation, epilepsy can take place. And carbon dioxide is responsible to maintain the level of acidity, and whenever you have a period of hyperventilation and a large amount of carbon dioxide is thrown out, the pH of the blood, it increases and it can cause an epileptic attack. Now what happens in acidosis? Opposite will happen. If alkalosis was causing overexcitation, acidosis is going to cause depression in the neuronal activity. So for example, the normal pH is 7.4, so depression, if it goes only to 7, what will happen? The person will have coma situation. That happens in diabetic or uremic acidosis, that these person have a large amount of acidosis taking place, the pH is decreasing and neuronal activity is depressed, and the person goes in the state of coma. That is how the diabetic patients become unconscious and we need to give them medication, etc. Now effect of hypoxia. Oxia is always related with the oxygen levels. Hypoxia means less level of oxygen. So whenever there is a synaptic transmission, it requires adequate supply of oxygen. So if there is no oxygen, there will be complete inexcitability. And what will happen? Synaptic transmission will be stopped. Brain's blood flow is temporarily interrupted. Like if it is interrupted for 3 to 7 seconds and we have a state of hypoxia, the person will become unconscious. That is the situation that we see in the case of Stokes-Adams syndrome, in which what happens is that heart cannot pump the blood to the brain. There's a state of low blood flow to the brain for about 3 to 7 seconds. Due to this, person becomes unconscious due to hypoxia. Now we come to the level of effect of drugs on synaptic transmission. Now I'll tell you this fun fact that whenever you are tired, what do you take? You take coffee, you take tea, you take drugs, which not drugs but coffee, tea, or black, that is dark chocolate. They contain caffeine, theophylline, theobromine, which are actually going to increase the synaptic transmission and the neuronal excitability. Now we have another drug which is strychnine. And what it does is, it does not decrease the threshold, rather it inhibits the action of inhibitory transmitters. The inhibitory transmitters that we studied, for example, you have strychnine. So the inhibitory transmitter channels that were opened first, now they are going to be stopped. Why they are going to be stopped? Because strychnine closes them, as a result of which there is no inhibition, only excitation, and the excitation causes you to have a large number of excitability. Now, due to this, tonic muscle spasm will also be produced. The other thing is that which inhibitory substances is strychnine stopping? Most substances are glycine in the spinal cord, and whenever strychnine will take its action, it will produce tonic muscular spasm. And now where there are anesthetics. What are anesthetics? The anesthesia. And whenever you are given anesthesia, what happens? You go to the state of coma. It means that your neuronal excitability is decreasing and you increase the threshold. Whenever you increase the threshold, what happens is that the membrane will become negative, more negative than minus 65 millivolt, as a result of which the person can go to a state of partial coma, that we can revive him. After that, the main thing about anesthetics is that they are lipid soluble and they can change the physical characteristics of neuronal membranes. That is, they can close the sodium channels, etc., and make them less responsive to the excitatory agents like the sodium channels. So anesthetics can change physical characteristics like changing the excitability or the reception of the sodium channels. Now, what is synaptic delay? Now, if you're familiar with this diagram of the neuron, you're all set with the synaptic delay. So what happens in the like in this principle is that whenever an action potential will come, for one thing, it will discharge the neurotransmitter. So neurotransmitter is discharged. Second thing, their neurotransmitter will diffuse into the synaptic space. First thing was discharge. Second thing is diffusion. Third, this diffuse now attaches to the receptor. Third thing is receptor attachment. Fourth thing is, due to this attachment, what happens is either sodium channels are opened or second messenger system is activated. That is known as the action of the receptor. And due to this action, a cellular response will be generated, that is, excitatory postsynaptic potential will be produced. So what do we come to there? Discharge over here, then diffusion, then attachment to receptor, then action of the receptor, that is activating certain ion channels or second messenger system, and third is and the fifth one is production of excitatory postsynaptic potential. The same thing is written over here, if you can read all of that. First thing was discharge, second thing is diffusion, third thing was attachment of the transmitter to receptor, fourth thing was action of the receptor by activating the channels or the second messenger system, and as a result excitatory postsynaptic potential is produced. But what happens over there is, due to all these functions, there is a delay of 0.5 milliseconds. So if scientists can actually measure the time that it takes for for a signal to reach the presynaptic terminal, that is input volley of impulses, and the output volley, that is its conduction to the axon. So the time period that is between these two, for example, they come at the level at 6.5 seconds, it comes over here, and at 7 seconds output volley is taking place. So the difference between the two is 0.5 seconds. This is termed as synaptic delay, and due to this delay we can actually measure the working of the neurons. So now we are done with the chapter number 46 of our neurophysiology. If you like this playlist of my neurophysiology, please do follow, do subscribe to my channel so I can make more videos, I can be motivated to teach you guys. I am forever thankful, forever grateful for the help, and for the help that I can give you, and for the help that you can give me in the form of subscribing to my channel. And also I have my Instagram page that you can also go to, so also support me over there till we meet till the next time. And if you have any questions or queries or any sort of recommendations or videos that you want me to make, please let me know in the comments below. Till then, Allah Hafiz, meet you in chapter number 47.